Hey everyone, welcome back to Windows C Studies. I hope you all are doing well. So we were discussing cellular signaling, in which we have covered the basic signaling pathways, how the cells are able to communicate with each other, right? So we discussed that how a cell needs to have a receptor protein so that it is able to receive the extracellular signal molecule. So this receptor protein, when it reacts with the ligand, this ligand receptor interaction is going to activate that particular receptor protein. So this activation of receptor protein could result into the cell signaling pathways, the intracellular signaling cascades, which could result into the effective proteins such as metabolic enzymes, or it could result into the transcription of transcription regulators or some cytoskeletal elements, right? So whatever the effect that signaling intracellular signaling cascade could have, that effect varies depending on which type of signaling pathway is being followed by the cell, and that signaling pathway depends on which type of extracellular molecule is being received by the receptor protein. So when this receptor proteins does the conformational changes in its extracellular domain, those conformational changes will be mirrored on the intracellular domain due to the transmembrane domain, right? So this conformational changes on in this receptor protein on the intracellular domain could activate the enzymatic domain which is present in this intracellular domain, or it could couple this intracellular domain with certain inactive enzyme, right? So it could be different. So what once this receptor protein gets activated, either it could directly switch on the molecular switches, or it could get coupled with some enzyme which is going to in turn switch on molecular switches, or it is just coupled with ion channel, right? So depending on those three choices, the cell surface receptor are categorized into three different categories: the GPCRs. Okay, so the three categories were GPCRs, the G protein coupled receptors, that we have discussed in detail. How do they work, how they receive the extracellular signal and result into different cellular effects, right? The second category is the enzyme coupled receptors, and the third category is ionotropic receptors. Okay, so those three categories, in these three categories, I told you that either the receptors could be coupled with the coupled with the molecular switches. The molecular switches, basically they are the proteins which could be switched on and switched off according to the cellular need, according to the conditions that exist in the environment. And when they get switch on and switch off, they have the ability to control the gene transcription, to switch on the gene or switch off the gene. So those molecular switches are the core of the signaling. Okay, so they are very crucial for the signaling to work. And we learned that there exist two different type of molecular switches: the GTP binding proteins, because hydrolysis of GTP is a direct switch. Okay, so if the GTP is being bound with certain protein, means that protein is in its activated form, and when this GTP gets hydrolyzed, it is going to do its downstream job, and due to the hydrolysis of GTP, it will be converted into the protein which is bound to the GDP now, right? So this GDP bound protein is in its inactive form. So GTP hydrolysis acts as a direct switch, and that is why GTP binding proteins are the major category of molecular switches. The other type of molecular switches are the ones where the certain protein gets phosphorylated. So phosphorylation of certain protein means that protein is getting activated, and now this activated protein is going to do its downstream job. So this activation by phosphorylation will be done by kinases, and those downstream signaling processes are known as kinase cascades. So kinases and phosphatases, both of them, they together act as a molecular switches so that the signaling works, right? So those two categories of molecular switches are important. So the receptor proteins, when they are being coupled with the G proteins, the GTP binding proteins, they are called as the GPCRs. So the G proteins could be of different types. Okay, the G proteins could be of they could be stimulatory G proteins, the inhibitory G proteins, the GQ proteins, the G12, G13 proteins. So we discussed all of them in GPCR lectures. So then there are other type of receptors known as enzyme coupled receptors. So instead of being coupled directly with a molecular switch, they are being coupled with an enzyme. Why does the cell choose this, right? Why is going with a lengthy process rather than just being coupled with molecular switches, just like in the case of GPCR? This is because when cell is being coupled with some enzyme, okay, let's say that it is being coupled with tyrosine kinase. Okay, so tyrosine kinase is going to add phosphate group over the tyrosines, resulting into the formation of phosphotyrosines. But this process is reversible. Why? Because this type phosphotyrosine residues could be dephosphorylated with the help of phosphotyrosine specific phosphatases, right? So whatever kinase is going to act on, that reaction could be reversed with the help of phosphatase. So this is a kind of short-term signaling. So this short-term switch is provided so that the rapid changes in the cell could happen. But for this short-term signaling to be able to convey long-term effects, to be able to show long-term effects, they are being coupled with G proteins. They are being coupled with the molecular switches. So you get it, why the cell is going with a lengthy. So it is a kind of two-factor authentication for the cell, or a two-way gate for the cell to ensure certain processes being regulated. So whenever we add more number of gates to any process, it ensures the better control. It gives us a better control over the situation. It gives us the control over the situation to be able to regulate that properly, right? And that is why the enzyme coupled receptors are being coupled with the enzymes, which could get activated when the extracellular signal arise. And this activation of this receptor will result into switching on some certain molecular switches. So we learned about, so the major category of enzyme coupled receptors, it is the receptor tyrosine kinases, right? So we started learning about receptor tyrosine kinases, and we saw that how they are being coupled with different types of G proteins, or different types of molecular switches. So in last class we learned about how RTKs, they are being coupled with RAS proteins. So RAS proteins, RAS families, they are the monomeric GTPases. So RAS is a monomeric GTPase, and thus on the RAS protein, when the Ras-GEF works on this RAS protein, it replaces the GDP with GTP, and that is how the RAS protein will be switched on so that it can perform its own downstream signaling. So we learned about RAS, how RAS activates MAP kinase module in last class, right? So the other type of GTPases that could be activated by receptor tyrosine kinases is the Rho family GTPases. So Rho family members, it is the subclass of the RAS superfamily GTPases, and there are different family members such as Rho protein itself. There exists CDC42. So let me just write it down here. So there are different family members such as Rho, CDC42. Okay, and there's one more called RAC. Okay, so these are the different members of Rho family, and they work in the different condition according to the different extracellular signal. So today we will be learn about Rho signaling in detail, how this Rho family of how this molecular switches Rho proteins are going to work downstream. Okay, right? And the third type of the receptors, which are not being coupled with any type of switch, they are just there so that the membrane potential could be changed directly, are the ionotropic receptor. So they are not they are not doing some downstream signaling through the help of any type of molecular switch. They are using the change in the membrane potential as a switch, right? So membrane could get depolarized or membrane could get hyperpolarized so that it could result into different signaling pathways, right? So this is really a nice way. So cells are the best designs. They are best machines which are present naturally, and look at how beautifully different receptors are there and different switches exist so that the gene transcription could switch on and switch off. Okay, so to summarize, GPCRs use G proteins as the molecular switches. Enzyme coupled receptors use the enzymes so that it could switch on the molecular switches such as RAS, Rho, okay, and there are many more that we'll be learning in consecutive lectures. And ionotropic receptors are the one which uses the membrane, which changes the membrane potential once the signal arrives by opening the ion channels, and this change in the membrane potential will act as a switch. So there's no molecular switch existing here, but this membrane potential change will act as a switch which could switch on certain signaling pathway, right? So we have discussed about this. We have also discussed about this. We discussed about inotropic receptors in neurobiology, and we discussed about GPCRs in the couple of lectures few days back, right? So today we will be discussing about Rho signaling in detail. We have discussed about RAS-MAP kinase pathway, and in the next lecture we will be discussing about PI3 kinase pathway. And with this we will be done with the cell surface receptor part. Okay, so let's discuss the Rho signaling in detail. So just now we learned that Rho proteins, Rho family members, they are going to work through the receptor tyrosine kinases, right? So the receptor tyrosine kinases, they are the cell surface receptors. So this is the plasma membrane, of which this is the exoplasmic leaflet, and this is the cytoplasmic leaflet. Okay. So you remember I told you that when the those receptor tyrosine kinases has three domains. Okay, so it has three domains: the extracellular domain, transmembrane domain, and intracellular domain. Each of them has its own job. The extracellular domain is the one which is going to receive the signal, and it is going to change the conformation. So whatever conformational changes are being done due to the extracellular signaling molecule attaching to this extracellular domain, the transmembrane domain will also have some conformational change, and that they will be mirrored in the intracellular domain. So this intracellular domain, what is the job of intracellular domain? Either it has enzymatic activity in its itself, so it has intrinsic enzymatic domain, or it could be coupled with some inactive enzyme, right? So there exist two possible scenarios, that it has the intrinsic catalytic domain, such as the one we discussed in last class, when it activates MAP kinase module, right? When it activates RAS family, in that case, the receptors, that receptor tyrosine kinases are the receptors which are receiving the growth factors. So EGFR, or the platelet derived growth factor receptor, or insulin receptor. So the receptors which are going to receive the growth factors, they have intrinsic tyrosine kinase activity. So they have intrinsic tyrosine kinase domain. But in the case of the receptors which are receiving the Rho family, which are receiving the Ephrin, so which are going to which are going to activate Rho family proteins downstream, they does not have intracellular catalytic domain. Rather, they are being coupled with the inactive enzyme. Okay, so they get coupled with the inactivated enzyme. So what happens is that the receptor tyrosine kinases, they received extracellular molecule which is in its dimerized form, and that is why it also catalyzes dimerization of the receptors. Okay, so this is the Ephrin molecule. Okay, so Rho signaling is also, we could say that the Ephrin molecule signaling is mediated through the Rho family members. So this Ephrin molecule is being received by the receptors. So that is why those receptors are termed as Ephrin receptors. Right, so this Ephrin molecule, when it binds to those receptors, it is going to promote the dimerization, right? So let's just draw a dimer here. So this Ephrin molecule, due to it is promoting the dimerization of the receptor. So those receptors, when they are being in the vicinity, they are coming in the close proximity, the inactive tyrosine kinases domain that are being coupled with them will get activated, and they are going to do the phosphorylation. Okay, so these are the tyrosine kinases. So first they were inactivated, so they were in the inactive state, but now they are being activated, and that is why a phosphate is being added on the tyrosine residues. Okay, so the phosphotyrosine residues will exist on there. So this activation of so this active, so the phosphorylation of those tyrosine kinases will activate them fully, and now those tyrosine kinase will work on the family of Rho-GEF. Okay, so there's a protein called Ephexin. Okay, let me draw this, which is being coupled here. Okay, which is attached to the receptor itself, and this Ephexin is nothing but a Rho-GEF. This is Ephexin, which is a Rho-GEF. Okay. You remember that in the case of Ras-GEF, it was being coupled with the plasma membrane with the help of lipid anchors, right? So lipid anchors were holding the Ras-GEF on the attached to the cytosolic leaflet, and when the tyrosine kinases, they were phosphorylating SOS. The SOS is it is the RAS-GEF. It was activating the RAS protein. So RAS protein was, it was held to the cytosolic leaflet with the help of lipid anchor. I hope you remember that. But in this case, the Ephexin, which is a Rho-GEF, which the response of the EPH receptors depends on the cell surface, so not the cell surface, sorry, to this Ephexin, which is being coupled with the cytosolic part or the cytosolic domain of EPH receptor, right? So this tyrosine kinases, they will work on this Ephexin, this Rho-GEF, and they are going to activate this Rho-GEF. So they are going to phosphorylate Rho-GEF, and it is going to activate Ephexin molecule. So this activated Ephexin molecule is going to do its downstream job. So this is a Rho-GEF, right? So this is going to what is the job of GEF? GEF is going to remove the GDP. It is going to remove the GDP and it is going to add the GTP. So it is going to replace the GDP with GTP. So GEF will, what will Rho-GEF do? So it will remove the Rho, it will remove the GDP which is being attached with Rho protein, and it will replace it with the GTP. So this is the job of Rho-GEF. Okay, so that is what Rho-GEF does. So this activated Rho-GEF means activated Ephrin molecule. It will replace the Rho-GDP with the Rho-GTP, and this Rho-GTP is going to do the downstream job. So let me show you a schematic from the Bruce Alberts. Okay, so this schematic beautifully explains the whole process. So here you can see Ephrin molecule is being received by the Ephrin receptor, and due to that the Ephrin receptor gets activated. So this Ephrin receptor, the tyrosine kinase is being coupled with that, and it gets activated due to the receiving due to the ligand receptor interaction. So this activated for tyrosine kinase, what does it do? It act So as I said, first the receptor, so first tyrosine, so this inactive tyrosine kinase, it phosphorylates the phosphotyrosine residues present on the intracellular domain, which fully activates the tyrosine kinase. Okay, and this fully activated tyrosine kinase, it is going to phosphorylate the Rho-GEF, okay, which is also known as Ephexin, which is present on the which is anchored to the intracellular domain. So you remember RAS-GEF was not anchored to the intracellular domain, but the RAS protein was anchored to the cytosolic leaflet. So in the same way as the RAS protein, there exists Rho family member known as Rho-A, and it is being anchored with the cytosolic leaflet. But the Rho-GEF, you remember the RAS-GEF, okay, RAS-GEF was SOS, son of sevenless, and son of sevenless was being anchored to the phosphotyrosine residue. You remember that? But here the Rho-GEF, which is also known as Ephexin, it is not being anchored to the phosphotyrosine residue, but it was actually coupled with the intracellular domain. Okay, so it is not associating itself with the phosphotyrosine residue. That is for clear. So once this Rho-GEF gets phosphorylated, means it is now activated, so it is going to replace the GDP which was present on the inactivated Rho-A, and this GDP will be replaced with GTP, and this active Rho-A is going to do its downstream job. So what does it do? This activated Rho-GEF, it is going to activate Rho kinase, and this Rho kinase will result into the cytoskeletal movements. So due to that, it could change the cell shape, cell polarity, cell motility, and even cell-cell adhesions. In certain cases, Rho kinase, the activation of Rho kinase will also result into the cell migration or nerve axon outgrowth, or it could mediate certain gene transcription, so which will help into cell progression, and which can also help in the membrane transport. So these are the different types of downstream effects that Rho family members, the Rho A protein, could have. So this is with this I conclude this lecture. We are done with Rho signaling. And in next lecture we will learn about the PI3 kinase signaling. And with the PI3 kinase signaling, we will be done with the whole signaling part. I mean the receptor, the cell surface receptor part specifically. And then we'll have a kind of revision lecture, or kind of a summary lecture, where we will be discussing all of the different signaling pathways that we have learned, and how those signaling pathways, they are interconnected with each other. So for example, that a certain extracellular molecule will activate certain receptor, and a single receptor can activate multiple downstream signaling pathways, or a downstream signaling pathway can be activated by different types of extracellular signal. So we'll learn about how this network of cellular signaling is very interconnected, and we'll learn about the PI3K signaling as well. So I conclude it here. Till then, see you. Bye bye.